Hi everyone, welcome to your very first presentation session 1. This session has an overarching theme, reproducibility in biomedical modeling, and we will have 4 10-minute presentations followed by a bundled question and answer session. If you have any budding questions during any of the presentation, please feel free to pop them onto the live Q&A on the right-hand side of your presentation session window. And we will address them one by one at the end of all 4 presentations. If you have any technical problems, please feel free to press the red live support button on the top right-hand corner of the screen. So without further ado, I would like to introduce our very first speaker, Dr. David Nickerson, who will do the session introduction.
Cool, thanks, Erin. Just see if this is going to work again. Okay, hopefully you can see my slides. Where's that gone? Okay, so thanks everyone for tuning in. I just wanted to briefly start and mention a bit about reproducibility in biomedical modeling. I think as we've all seen with the recent pandemic, sort of modeling in biology and medicine has been quite focused in the media. We're seeing lots of issues being raised about credibility. And obviously reproducibility is key to how we're trying to address these. So I just wanted to briefly, before we get into these talks, mention a couple of projects that I'm involved in that kind of show how in the sort of biomedical realm we're starting to catch up to some of the other fields that have been focused on reproducibility and addressing things like this for a long time, where we can start to see a lot of progress. So the projects I'm going to talk about are funded through the US National Institutes of Health, but I think there's a number of agencies now around the world where reproducibility and things like FAIR data are becoming important. So the first one I just to briefly mention is the Center for Reproducible Biomedical Modelling, which I think fits quite nicely to this session. It's all about reproducibility in biomedical modelling in this website. And you can get these slides via feature if you want later. So in this center, it's kind of split into a few different groups, a couple based in the University of Washington in Seattle, one in Mount Sinai and Yukon Health, and the Auckland Bioengineering Institute. And it's split into 3 parts where we really want to look at the reproducibility of biomedical modeling from sort of inception through to the end product. So it's all about how do you reproducibly build models from data, how do you reproducibly simulate model and get results, and the sort of annotations that underlie how that can be done. And then there's also a big component of training and dissemination. You know, obviously there's a lot of these things that get developed and how you get people to use them and teach people what they should be doing. And that's actually led by a graduate student in Seattle, Veronica, who's doing a lot of work in terms of putting together tutorials and online material. But also part of what we do in the center is trying to work with journals. So obviously, in academic research, you know, it's all about journal publications. And so if we can help the journals improve the way they address reproducibility, we hope to then encourage, you know, people to change practices and adopt better practices to make sure that their modeling work is reproducible, which given it's computer code, generally it should be reproducible. So with this curation service, we're trying to work with journals to try and improve the way they do reproducibility and how we can address that. So just to mention briefly, we have been running over the last few months a pilot with PLOS Computational Biology, where we try to fit in essentially a reproducibility review as a technical assessment when people submit articles. So it's essentially designed to fit in the traditional peer review workflow where manuscripts get submitted and they get sent to us for this sort of technical review. And we sort of look at it to see whether or not this work is reproducible. And over the last few months, it's a sort of voluntary opt-in process, and about 70 authors chose to opt in. of which we've reviewed sort of 40 or so, probably a few more by now. And of that, 30% of what we call reproducible, and that at least one of the reported results could be reproduced. Often it's not all the results, but of that you can still see 30% is quite low for these people who are opting in to say that they think their work is reproducible. And there's a bunch of reasons we see why. So then just to shift gears a little bit, the other project I just wanted to briefly mention is the SPARK project, Stimulating Peripheral Activity to Restore Conditions. It's sort of looking at how you can use neuromodulation to improve human health. And this is a massive project, and it's quite different in that they mandate and require fair data sharing. So all data produced and things produced in this project have to be shared, have to be open, have to, obviously be reproducible. So just to give an idea of the sorts of things we're doing, this is where you're collecting data from many different specimens and you want to map them to these sort of common frameworks so that you can reproducibly understand what data people are collecting, how that is used, and then eventually use that in models. And this is supported through this data resource center, which again the Auckland Bioengineering Institute is part of. And it's all about trying to make sure that everything collected in SPARK is fair. Not sure about time, but hopefully it's going. Just to give you an idea of scale, in this sort of data submission and publishing workflow that's been set up under SPARK, there's now around 550 members and 25 terabytes of data. Just quite a good chunk. And because this is all required to be fair, it's not, the funding itself is contractually tied to people sharing their data. I'm not allowed to do that. I will share our data afterwards, or once we publish it, it has to be shared as we go. And then part of that is trying to make this information and this knowledge available on the web, which is quite important. So we're also putting together this portal across the SPARK project to try and enable this so that people can both find, access, and reuse the data. And as we go forward, more and more of this is bringing into the modeling and simulation workflows to see how they can be done. And of course, it's all got to be done reproducibly. So that's just a couple of projects I wanted to mention. And then I think for the rest of the talks in this session, we're going to see some actual examples of this in practice, looking at some of the tools being developed into how we standardize, how we can share and exchange models in reproducible ways, how we can use annotations to help with findability and accessibility and reuse, and then finally how we can help try and encourage better publication of reproducible models. So I know that was quick. The slides are available if anyone would like them. And I can't see anywhere what sort of time we're at. But that's pretty much what I wanted to say. Thank you.
Thank you very much, David. You had a few minutes left, so I think we'll relay that time to Hugh, if you don't mind. So we'll just continue on to the next session by Hugh Sorby, talking about reproducible and FAIR modeling with CellML and libCellML. So Hugh, whenever you're ready. Hugh, do you mind unmuting your microphone, please?
All right. Can you see my slide?
I can see your slide and hear you clearly. Thank you.
All right. It's got a bit of housekeeping to do on my screen. All right, right, I'm ready to go. I am going to talk about CellML and libCellML and the part they can play in reproducible and fair modelling. Did my screen just change then? My screen sharing was paused. That doesn't seem good.
Yes, can you please click resume screen share little tick box on the top?
I've got to find it. I've got a lot of screens open.
Do you mind refreshing the screen then? Because we see the change.
Okay. All right. Right, sorry, I skipped this page there. Right, so I'm going to talk about this in the context of computational biological modelling. And this will probably come as no surprise to you when I say this, but the modelling we are primarily interested in is cell modelling. Cells that we find in the human heart, for example. So of all the computational biological models that we find in literature, we can reproduce about 10% of them. That means if my maths is correct, we can't reproduce a staggering 90% of the models we find. So why can't we reproduce these models? Part of that is being able to reproduce the output found in literature is really important for the confidence that we might have in the robustness of the presented work. And another follow-on question is, how am I to reuse a model that I cannot reproduce? So some of the reasons why I can't reproduce a model is like I simply can't find the model. Often we have an incomplete description in the literature. We may only see part of the model in the paper. I might not be able to access the model. It has not been shared, or I don't have authorization to view the model. I can't interoperate with the model. Let's say, for instance, that I'm a Windows user, but the model was written by someone on a Mac, and he uses the fork command. As a Windows user, I don't have that functionality available to me. All of these stop me from reproducing what is given in the literature. And again, if I can't reproduce a model, how am I going to reuse it? So if I can make models fair, models that are findable, accessible, interoperable, and reusable, I eliminate a lot of the barriers to reproducibility. One of the easy things I can do to make my model reusable is to make it open source and make sure it is licensed appropriately. This takes virtually no effort. To make things reproducible, I need some tools. Computational biology is a heavy reliance on software, which also helps us to make things easy to reproduce, if they're done properly, and if only there were standards to follow. But there are. CellML and libCellML are two parts of the ecosystem that can help us reproduce computational biological models. But there is more to it than just these two parts. This ecosystem is made up of standards and tools, which I'll talk a little bit about now. So CellML is a founding part of this ecosystem for reproducible models. It's an open standard, meaning anyone is free to implement it. CellML is a specification that describes how a model information is written in XML. It is clearly focused on biological models, but it does have broad applications. Not in and of itself is not something that we want to interact with. We need tools that do that for us. And CellML is part of Combine, the Computational Modelling and Biology Network, which is an initiative to coordinate the various standards and formats for computational models. Here is some sort of visualization of a CellML model. The green container represents the model, the grey circles represents units used in the model, the blue parts represent components of the model, and the sigmas represent math that really holds the model description, and the yellow circles represent variables in the component used by the math. One of the things that CellML does is define interfaces to help make models reusable. Variables in a component are defined such that you can interact with the model in a defined and controlled way. It also defines imports, which allows modelers to reuse model components. If you have developed a potassium transporter model, I can import that and make use of it in my own model. Having units explicitly stated helps make the model reusable. If you define your membrane potential in millivolts, and I define my membrane potential in volts, we can still have our models interact and not be surprised by a scaling difference of 1000. This is just a simple example, but it really helps us to, when we units of an entity are not what we may assume them to be. libCellML is another part of this ecosystem. It is an open source software project, and its primary aim is to implement the CellML2 specification. It is a rewrite of an older software project, the CellML DOM API, into a more user-friendly form that is not based around the XML DOM, but CellML objects. It is written in C++ with bindings for Python and JavaScript, and it gives functionality to manipulate CellML2 models. It is available across all the major operating systems and some of the minor ones. libCellML follows software best practices. It also follows a test-driven approach. Given that it is a library, this type of development is particularly suited to libCellML. libCellML uses BuildBot for continuous integration, where the code base is tested on all operating systems that it officially supports. Also, because libCellML started from scratch and not from a prior code base, it is able to demand 100% coverage of code within its unit tests. Whilst we cannot say we are bug-free, we can say how the software behaves when the given stimulus that we test with. With 100% coverage, we also get help on resolving bugs when we do find them. And of course, we can avoid regression. 100% coverage is just one part of the acceptance criteria for new code. We also have memory checking and code formatting checks. We also have a code review process, and we're always looking for volunteers to review our code. A big part of a successful open source project is the quality of its documentation. libCellML places an emphasis on documentation. It is the first point of call for developers looking to make use of it. What sort of things can we do with libCellML? Well, we can create and edit models and other fundamental actions like serializing and deserializing. Importantly, for creating reusable models, it provides a validation service that validates the CellML model against the specification. libCellML provides an annotation service so we can make elements of the model accessible. It also provides a code generation service for turning a declarative model into a procedural one. This allows insertion into simulation software like CVODE. What are the other parts of this ecosystem? Well, PMR, the Physiome model repository, is an online platform for the storage of models. PMR can house any model, but again, primarily biological. PMR allows our models to be found and accessible. CellML is part of Combine, the simulation experiment description language. This gives us the ability to describe how our models are run, what inputs it requires, what model it uses, what outputs it should give, right down to the plots that we may generate. CellML allows for our models to be reproducible. OpenCore is an open source cross-platform modeling environment for the desktop. For our purposes here, it allows our models to be reusable, but it does do much more. OpenCMS is an open source cross-platform modeling environment for HPC environments. This gives us access to high performance computing that allow for multi-scale simulations where we can run whole organ model simulations right down to the cellular level. And CMGUI is a visualization application for viewing the results from OpenCMS. So I'd like to thank these agencies for their support in producing this work, the Auckland Bioengineering Institute, the Virtual Physiological Rat Project, and the MedTech Core. Thank you for listening.
Thank you very much for the presentation, Hugh. You were bang on the 10-minute dot. Thanks for keeping well in time. Please feel free to pop questions as we go to the live Q&A. We already had one question that has been answered. Next up, we have Dewan Sarwar doing a presentation on epithelial modeling platform.
Thank you, Erin.
Yep, whenever you're ready.
Yeah, I'm ready. I'm going to share my screen now.
Yes.
Can you see my screen?
Yes, we can see the title slide. Thank you.
Thank you. Hello everyone, good morning. I am Dewan Sarwar. I'm a research assistant at the Auckland Bioengineering Institute. Today I'm going to talk about on a research-based software platform, Epithelial modeling environment that uses reproducible computational models from the Physiome model repository. So the motivation of this tool is that given a collection of mechanisms and or observations, such as imaging data or diseases from reference ontologies, biomedical ontologies, or drug actions, or clinical observations, and so on, then can we extract a computational model from the Physiome model repository that will help to investigate clinical or experimental hypotheses? So as you can see here, this is an overall architecture, and where the epithelial modeling platform, that is the main topic of today, is in the middle. So on the left side, there are two simulation environment, OpenCore and SemGen. So these are the mathematical modeling environment. So we can edit mathematical models and deposit the models in the Physiome model repository, which is on the top right corner. And from the epithelial modeling environment is a web-based interface where we can discover and explore the deposited model in the PMR via some web services from the European Bioinformatics Institute. So there are three steps of this process. The first step is semantic annotation. First step is model annotation. So the model, I mean that by using model annotation, we describe the mathematical models parameters with biological meaningful information. The second step is semantic annotation using a tool called SemGen, that we put the biological information in the SemGen tool, and SemGen creates metadata, and then we deposited this metadata in the Physiome model repository. And the third step is to we have developed web interfaces to discover and explore those metadata from the Physiome model repository. So as I mentioned earlier, the first step is model annotation. In this step, we extract the mathematical models parameters from the reference published articles and describe them with a biological meaningful information. For example, if we have a variable, for example, J_Na, then I describe that variable with, for example, concentration of sodium in the cytosol compartment, and so on. In the next step, as you can see here, this is an interface of SemGen tool. So we put the biological information in the SemGen tool and choose some reference ontologies from the tool. And then this tool creates RDF metadata in the RDF metadata, and then I put this metadata in the Physiome model repository. In the final step, and we developed a web-based platform, epithelial modeling platform. So the user will come along on this web-based platform and will search some queries. For example, in this case, flux of sodium. And as you can see here, there are some informations is displaying. For example, in the model entity, there are some CellML variables and the biological meaning that we annotated in the previous step. And then the species of this model during the experiment of this model, and then gene and protein names. So the user will search some queries here, and then we'll choose some models from this list. And then we'll save the list of models in this page, and user will go back again and search a new query, and will choose some models, and then again go back to this step here. So this step is a list of models that will help the user to create a new model. So from this list of models, our web-based interface will visualize the models like so here. So this is a computer representation of a cellular environment. So as you can see here, this is an epithelial cell, and this is another epithelial cell in the middle. This is the paracellular compartment, which is indicated on the top right with a unique color. And this is this compartment. These are the rectangular shapes. Boxes are different compartments in the cell. So this is just a visualization of the models here. And the circle with the arrow sign represents the fluxes, and the polygon shape represents the channels. But in the background, for each of these circles and polygons, there are some required parameters that will help to create a new mathematical model, as you can see here. So this is the first object, which is the first circle in the previous page here. So here, all the relevant terminologies and URIs have been stored to create this new model. In the final step, we composed these fluxes and channels into a new mathematical model using libCellML that Hugh Sorby described in the previous presentation. So the end goal of this model composition is to produce a CellML model that will help to investigate experimental or clinical hypotheses. I would like to thank David Nickerson, Karin Lundengård, and Peter Hanser for supervising this project, and thanks for funding MedTech and Auckland Bioengineering Institute. Thank you. Now I would like to pass my microphone to Erin.
Thank you very much for the presentation, Dewan. We are keeping very well with time. I would like to advertise again, if you have any questions, please feel free to put them on the live Q&A. And we're on to the last speaker, Dr. Karin Lundengård, talking about Physiome curation system for reproducible models. So Karin, whenever you're ready. And I can confirm I can see your screen.
When I started sharing my screen, the mute button disappeared. Can you hear me?
I can hear you.
Okay, very good. Thank you. So my name is Karin, and I'm here to represent scientific journal Physiome that we have recently launched. And it is a journal about reproducible mathematical models of physiological processes. And because these models are implemented as computer code, one would think that they should be very easy to reproduce because, I mean, it's code. You should just be able to send it to someone else and they could run it. But anyone who's tried to use these models, just as Andre and Hugh has been talking about, will realize that this is not the case. You get a lot of errors and it's really, really hard to get someone else's model to work. And not only is that intensely frustrating, but it does affect our scientific output. So the Center for Reproducible Biomedical Modelling that Andre was talking about, or David was talking about, they looked at 650 curated models in the two repositories, Biomodels database and the Physiome model repository, and realized that 90% of these models could not be reproduced on initial attempt based on the published information by an independent group. And some of these models even couldn't be reproduced by the group that published them in the first place. So even if people try to make the code available, that doesn't mean that these models are reproducible. And this is what Physiome is trying to fix. So we help authors make their models available in appropriate formats by publishing curated and version-controlled code. And the reason why we are in the format of a journal and not just a repository is that we want to give credit where credit is due. Very often in like the classical standard way of publishing research, the modeling, the implementation of the model doesn't get much space, which means that it's hard to get citations for your model. And that means that we lack incentive to actually make sure that the model code is reproducible. And of course, that also means that you get less money to support that, even if you want to make it reproducible, it's not in the budget. And of course, we want to publish this open access because we want it to be reusable for other people and in the framework of FAIR. So this is a Physiome publication, consists of 3 different parts. The most important part is, of course, the code. But then every Physiome publication is connected to something we call a primary publication, which is an article in a field-specific peer-reviewed journal. And that's where the scientific validity and the validation of the model and the mathematics are presented, and then we publish the code. And then there's also a manuscript, which is a descriptive summary of how to run the model and what code is included. We have 4 different types of publications. What I just described is the original Physiome publication. You can also publish a letter, which is a minor change to an already existing model, or if you've found an error and you want to correct it. A retrospective is pretty much like an original article, but it's if you take someone else's model that maybe was published 20 years ago and you update it to a modern standard and make the code available. And we also publish reviews. We can give an overview of the modeling of your specific field of interest. Everything is published on Figshare and is open access for anyone. So basically what we're saying is that there isn't really any good way of publishing code, and that's what we want to do. We want to make a new publishing model for physiological modeling. And by making the code and the model implementation citable, we will make it more discoverable, which in turn will make it more reusable because people can find it easier. When it's curated and version control, it's reusable or reproducible, which also supports reusability. And we also want to make standards and strategies for increasing modularity and annotation in models. And these 2 are strategies for if you want to combine 2 different models with each other, what standards should we have for that? And all of these will then lead to supporting reusability of the models. This is our webpage. If you want to read more about our philosophy or anything else about Physiome, welcome to have a look at it. And if you want to publish something, we have a manuscript template that Overleaf has kindly set up for us. And we have also, of course, a submission system where you can submit your articles. And in this system, the authors can submit, we can track the curation process and publish through the same system. And if anyone is interested in looking at how that works, the code is available open source on GitHub, and it's provided by a company called Digital Science, and is based on the COCO Foundation's system. So thank you for that, Digital Science. In the future, what I would like for Physiome to do, I have 2 dreams that I'm pushing within Physiome. And the first one is something that we call simultaneous publishing, where if you submit a model to a primary journal, you can also submit your code as a Physiome article, a Physiome submission. The primary journal would then do their normal peer review, and they can use us to curate the code. So we curate it and we send a curation report to them and tell them that this code is running and reproducible. Then they make a decision about whether or not they want to publish. If they publish, we also publish. If the code is up to standard, of course. If they don't publish, we don't publish, because then you don't have a primary publication. We don't want to compete with these primary journals. We want to be a complement instead. The curation as it stands now is that we are curators. We're working together with the Center for Reproducible Biomedical Modelling, and we look at the results that are presented in the Physiome manuscript and the results that are presented in the primary paper, and make sure that they match what the output of the code is. And we have a look at making sure that there's math present in the code as well. What we want to do in the future is that we'd like to run the curation process through something called Gigantum, which is a kind of a Docker where the authors would be able to run their code, and Gigantum would then produce a curation report that we can look at. And this would enable us to curate models that we cannot presently run on our system. For example, models that needs a supercomputer, etc. And my second dream is connected to curation. Right now we are 2 curators, and we are of course a bit limited in what kind of programming languages we know and what kind of models we're familiar with. So what I'd love to do is to set up some kind of platform or portal where we can have crowd curation, where people from all over the world can contribute, curate each other's models, and this would then end up in Physiome articles. And maybe it would be possible for experimentalists who are not familiar with modeling to go into this platform and discuss and ask, I want to do this, what models should I use, and get a deeper understanding of how to do this and maybe find collaborations, etc. This is the editorial board and the executive committee, the people that made it happen. And of course, we were also supported by several organizations, for example, VPH Institute and the International Union of Physiological Sciences, IUPS. And most of us are present at Auckland Bioengineering Institute. So to summarize, because I don't know how much time I got left, we publish reproducible mathematical models of physiological processes. We aim to increase discoverability, reproducibility, and reusability of these models because we believe that this will increase the quality of our scientific output and decrease the amount of frustration in the modeling community that we are in. Thank you so much for listening, and I hope you'll go and look at our webpage and be motivated to come publish with us.
Thank you very much for the talk, Karin. Looks like a very promising journal and very useful. I hope it takes off. So this concludes the presentation part of this session, and we will go into the question and answer. So we already had a question that's been answered. Just in case you haven't got a chance to see it, it's in the answered questions section of the live Q&A tab. Alexandra asked, what incentives do you have and you think there are for journals to improve reproducibility? And the answer said, we are starting to see evidence that papers describing reproducible models are cited more often, leading to greater impact, which journal publishing companies like. Also, a lot of editors know there are issues and are very keen to address them. They just need help to know what to do and how to integrate into long-established publishing practices. So I think this fits really well into what you're doing, what you guys are doing with the Physiome. So we have about 10 minutes for a live Q&A session. So I'll keep an eye on that. Oh, something came up. James Cooper. With regards to open curation, do you have any ideas yet on how to avoid negative curation, i.e. either people who are not sufficiently technically sophisticated or have malicious intent from engaging in curation?
I mean, this is of course something that we have to think about on the platform, and I think that in the beginning, the start would be that we have people that we trust who are set up as curators, and setting up standards for how to work. And then one thing that we also do in the journal now, because it might be that we can't run the code on our first try, and we have a lot of communication with the authors on how to do this, and we could make that communication open as well. So the communication between the author and the curator. And I think that if you have malicious intent, it's hard to hide that in such an open conversation.
So I'll archive that into the resolved answered question section for now. I have a following up question on that. So in order to run and check the validity of the code, you probably need the raw data that they use to run the code and solve the problem. Some of these data might be sensitive data ethically, like limited access and those things. How would you access those kind of data to check the validity of the code? Anybody from the panel?
So I can have a go at answering that. And yes, that's definitely an issue. And we've already seen that in a pilot with PLOS computational biology, where, you know, you can make all the code available, you can tell people how to use it, but without that data that they use to run their model, you can't reproduce results. And sometimes, as you say, you just can't get that data because of ethical reasons or whatever. So the way we see it is, you know, what you can do is you can provide test data. So you can demonstrate that your code is doing what you say it's doing, that your model's giving the right results for sort of well-known test data, which obviously isn't going to necessarily match or be even close to the real data. But if you can do that and demonstrate that your code is working and doing what it says it should do, producing results that, you know, an independent reviewer or curator can do, then that's probably as good as we can get. There are other efforts underway as well where you can sort of be able to access sort of sensitive data through various methods to hide it and obfuscate it and things like that. But that's probably not so easy. Thanks.
Thank you very much for that, David. There's another question just popped up. Does the platform require users to follow best practice, such as coding standards, tests, documentation, etc., for a submission? I think this is for Karin.
For our submission system, I mean, the curation is done by people. So the like our platform that we got from digital science has very few built-in checks. So all the checks that we do are things that the curators run manually, and this is because the type of models that we check are so diverse that it's very hard to set up anything automatic. But we have a checklist in it so that the curators have guidelines for how to check a model, and then you can go into that list and you can go, yeah, this model can do this and this model can do this, because not all checks are relevant for all models either. So we needed to have a really big flexibility, but that also means that we have a very like that the software that we're using is very open to a lot of different alternatives. Did that answer the question? I don't know.
It sounds good. Yes, I do understand there are so much options, so you can't have like a one manual to rule them all, but it'll be really nice to have some kind of a general guideline evolve for people who are new into this reproducibility field, to just have it as a general pointers as we go.
We are working with the Reproducibility Center, and we are working together with the Combine community as well to make recommended standards. And right now we have guidelines on our webpage for author instructions on what your model should include and what we need to be able to curate it, etc. And I'm going to get more information about where to find standards and how to find things that applies to your specific type of modeling in the future. But right now we have the author guidelines that you can go and look at, because there's quite a lot of information in that.
Thank you very much. I guess I'll ask another question. For CellML and those kind of things, can it be easily like translated into for other projects? Like this is a highly specific way of curating data for specific project, but can it be adopted for some other unrelated projects, do you think? Dewan or Hugh?
I'll have a crack at that answer. Yeah, I think so. I think a lot of people get put off by the name in the title of these projects. They think they're just for cells, or CellML, but this really isn't the case. They're for mathematical modeling, and they can apply to any sort of way, any model you can describe with your maths. So people are using CellML to do some COVID-19 modeling, for instance, and that is not to do with cells per se, but more to do with the movement of the virus. So it does have applicable to other areas, but the name, I think, fights against that.
Thank you very much. It's good to know that it has a lot of potential. We have 3 minutes left for this current session, so this is probably your last shot at asking any question. If not, I would like to ask for the entire panel, what was the biggest challenge in implementing reproducibility in your group? Is there something, if we would like to enforce reproducibility in our own research groups and we're not used to that, is there any tips or things to watch out for?
I mean, what Physiome is trying to do is to enforce reproducibility in all groups everywhere in the world by providing the incentive of citations. So, for example, our retrospective articles, I think, is a great way to get PhD students to get started on a model, because if they find a model that they want to use, they can implement it, and then they can make a Physiome article. And we really have tried to make it as simple as possible to do these articles. It's really easy to write a Physiome article. It doesn't need to take that much time as an original article or in a primary paper where you have to do all the work for experiments, etc. So it's a really good first publication, I think, for a PhD student, for example, and that is something you can use.
Okay, thank you very much for that.
So maybe just to add on to that, you know, the idea of reproducibility is good, but we've had tools and technologies around for a long time now to help address this. And what we find is, because it takes time and effort, right, to go the extra step to make your model available in a standard format, make sure it's reproducible, that someone else can use it. So I think a lot of what we're doing now is trying to make it easy for people to do. So it's easy for you to bring these types of tools and technologies into your existing workflows, which I think is, you know, is a real power of the Center for Reproducibility, because we're essentially funded to try and make these tools, make them accessible, make it easy. Obviously, it's a hard thing to do in general, so we focus on specific domains. And then the other side is trying to make reuse of models easier. So the tool like Dewan presented, where you can have a nice web interface where you can just use these types of things to build a model, which, because it's based on standards and reuse and reproducibility, you yourself don't need to worry about it, but it just, the product you get at the end of it is a reproducible and reusable model. So trying to do that, but also trying to do that in a way that's more general. So in a very specific domain, it's possible, but the general project is sort of ongoing.
Cool. Thank you very much. I think that wraps up the session very nicely. Thank you very much for all the presenters. If you have any questions regarding this presentation, you can contact them, any of our presenters, through our meeting hub. If you're on the timeline, just click on this session, and then you can see their contact details on the right-hand side panel. I think within 15 seconds, we'll be called back out, and this session will terminate. I'll see everybody back in the main keynote session, which is coming up next after the morning tea. 3, 2, 1. Bye.